In this video, we'll look at the serna paper. The serna paper begins with a description of a screen they carried out to find mutations in the system that regulates stomatal opening and closing in response to changes in carbon dioxide level. Now, in order to carry out this experiment, which must have been expensive, they had to hypothesize that if leaves are opening and closing stomata in response to carbon dioxide, then those leaves are going to be of different temperatures, because when stomata open, more evaporation happens. More evaporation leads to more cooling. Okay, so they write a grant. The hypothesis is if we look at plants in high, medium, and low carbon dioxide levels and expose these plants to pretty warm conditions and then measure them in pretty cool conditions, the residual heat of the leaves will reveal which plants had stomata that were open in all conditions or closed in all conditions, and which regulated stomatal opening normally. So you see that in a wild-type plant, when there's low carbon dioxide, leaves are cooler, see it's blue, than in a high environmental CO2 level. So here the CO2 level is high and the leaves are relatively warm, exposed to the same temperature. The only variable here is how much carbon dioxide was in the environment while we grew these plants. So here you see that they're warm because they're yellowish reddish. Here they're cool because they're blue. And cool plants are cool, these people postulate, because their stomata are wide open and evaporative cooling is reducing leaf temperature. So they took thermal pictures of hundreds of thousands of Arabidopsis plants and discovered a plant set in which no cooling occurred even at low carbon dioxide levels. Low carbon dioxide leads to open stomata because when carbon dioxide is limiting, you open the stomata to a larger extent, letting more carbon dioxide in. When carbon dioxide levels are high, it takes a less open stomata to get enough carbon dioxide through, right? The rate of carbon dioxide entry is a function both of the resistance, which is how big a hole it has to travel through, and the concentration driving the carbon dioxide in. So when the carbon dioxide concentration is high, you don't need a very big hole. When it's low, you do need a very big hole. Okay. What they discovered was a set of mutants, but we're only going to look at one, the HT1-2 mutant plants. And in these HT1-2 mutant plants, in every condition, the stomata stay relatively closed. Now, it's not perhaps evident from this picture that this level and this level are statistically the same, okay? So what this graph is telling us is that HT1-2 plants in all environmental conditions look just like wild-type plants do in high carbon dioxide conditions. So this is when HT1 isn't functional. This is when HT1 is functional. What you need to decide is what is the function of HT1? Is it involved in opening stomata or closing stomata? This is what happens when HT1 is missing. This is what happens at high, medium, and low carbon dioxide conditions when HT1 is present. So what is HT1 doing when it's working? Under what conditions is HT1 functioning? Is it functioning at low carbon dioxide conditions or at high carbon dioxide conditions? And to figure that out, you say, well, how do I compare it to when there's no HT1 present? This is what it looks like when there's no HT1 present. This is what it looks like under some conditions when HT1 is present. The next set of experiments the serna team carried out was to determine what part of the HT1 mutant alleles was broken. So they divided the gene, the broken allele of the gene, into two parts, the regulatory region and the coding region. They took the regulatory region and ligated it together with a wild-type version coding region. So this came from a wild-type gene, this came from the mutant gene. Now, remember, the mutant plant has hot leaves in low CO2 conditions, whereas the wild-type plant has cool leaves in low CO2 conditions. Because the stomata are open in wild-type, they're not open in the mutant. Here's another picture of the mutant, same picture. Now here is a picture under those same conditions of what happens mutant plant into which you add this construct, this hybrid between a regulatory region from the broken allele and the coding region from a wild-type allele. And look what it does. It doesn't look like the mutant phenotype anymore. It looks now like the wild-type phenotype. So ask yourself, given that if you throw into a mutant plant this construct with a wild-type coding region and a HT1-2 regulatory region, and that restores wild-type function, what part of the HT1-2 mutant allele is broken? Is it the regulatory region or is it the coding region? The next set of experiments that the serna team carried out were designed to reveal where in the regulatory pathway HT1 functions. Remember that when there is a reason to close stomata, now I've given you abscisic acid levels as something that closes stomata, but we can see too from this set of experiments that when carbon dioxide levels rise, stomata need to be closed. Closure of stomata by HT1 would take place either before the proton pump is slowed down. Remember, it takes slowing down the proton pump to depolarize the guard cells. When those guard cells are depolarized, the potassium channels open and potassium leaves. The chloride channels open and chloride leaves. This solute leaving leads to water leaving, and the stomata close. If carbon dioxide levels fall, or if there is blue light, say for instance, stomata open. What leads to stomatal opening? Well, the proton pump is activated, is cranked up, is turned to a higher level of activity, and that hyperpolarizes the cell. And that hyperpolarization opens a different set of potassium channels that let potassium in and activates a proton chloride cotransporter. So these ions come in, water follows. That puts pressure on the walls of the stomata open. So here's a paired negative feedback loop way of looking at stomatal opening and closure, but in this case, I'm telling you that homeostatic carbon dioxide levels are one of the features, one aspect of what dictates how open or closed the stomata should be. Okay, and we know that HT1 is involved in this process, but where? We don't know if it's occurring before the proton pump is activated. Maybe it's involved in the regulation of the proton pump. Maybe it's involved in the pump's opening and closure of channels and cotransporters. Maybe it has something to do with post-channel opening or closure, post-cotransporter activity. How do we figure this out? Well, there is a fungus that attacks plants, and the way that it attacks plants is to crawl through their stomata and, steal all of the goodies once it's in that spongy mesophyll. So it attacks plants by entering their leaves through stomata. Stomata can close to avoid this. Sadly, this particular fungus that makes this substance fusicosin makes the substance fusicosin to open stomata. So it inserts fusicosin, this molecule, into the guard cells. When it does, the guard cells open because fusicosin directly activates proton pumps. The serna team measured control, no fusicosin added, and fusicosin added wild-type plants and mutant plants. Notice that the measurements have been taken at low carbon dioxide concentrations. How do you know that? Well, because you can see that the controls for the HT1-2, well, that's low opening. But in the wild type, it's high opening, and wild type plants only open their stomata when there's low CO2. So low carbon dioxide conditions, then you add fusicosin. In both the wild type and the HT1-2 plants, you now have a maximally open stomata. So I'm asking you to assume that the statistics carried out by the authors of this paper are true and bear out the fact that there is no significant difference and that in both cases fusicosin fully opens stomata. If that's true, if fusicosin opens stomata, whether or not there is a functional, so whether or not there is a functional version of the HT1 protein, which of these two models is borne out by this data? It's either possible that that the proton pump is activating HT1, which is then opening stomata, right? When HT1 is active, stomata open. It's also possible that HT1 is activating the proton pump, which opens stomata. Either one of these is possible from the data shown before this set. This set helps you distinguish which of these two is possible. In other words, you can eliminate one of these models using this data.